Let's look at how we do a test cross. A test cross helps us find the original parents or the original organism. When you do a test cross, you always cross with a homozygous recessive. So for example, if you have flowers and you have a particular flower and you notice that that flower, that genotype, you don't know what the genotype is because this flower is actually purple. Now, if you have specific traits or specific genotypes that purple is dominant and you have a homozygous dominant for purple, but in this case you could also have a heterozygous because big P is dominant. So you don't know which organism or which is the original organism. Is it homozygous dominant for P or the heterozygous here for P? So you do a test cross. And what you do is you cross it with a homozygous recessive. Say this flower, for example, here gives you a white. So to cross with a recessive, homozygous recessive, you would cross it with each one of these possible individuals. So do that as a monohybrid cross first. So you cross in this P, homozygous dominant with homozygous recessive. So all the possible combinations would give me the heterozygous plant. All of these here would give me purple flowers. So if the original parent was homozygous dominant, all of my offspring, all of my progeny would give me purple flowers. So that's how I would know my parent is homozygous dominant. Conversely, if we cross with homozygous recessive, the combinations that we would get here is 50% would be purple and 50% would be white. If you get 50% purple and 50% white, then you know for sure that the original organism, the original plant, was the heterozygous condition. So that purple actually was the heterozygous plant. If you get 100% purple, it's the homozygous plant. And that's how you do a test cross, by crossing with a homozygous recessive.